Okay, good morning, everyone. Welcome to webinar number 21. Today we have Dr. Vinod Scaria with us. He is a principal scientist at CSIR-IGIB New Delhi, and his team works on understanding the function, organization, and regulation of the vertebrate genome and how genomic variations can impact the human population. So before we begin, we'll just do a short introduction about our organization, the Center for Predictive Human Model Systems.
Hello, everyone. Good morning. I am just going to share my screen. Okay. Hello, everyone. So the Center for Predictive Human Model Systems is a science and policy center and a think tank established in 2019. And our main focus is to enable new technologies and model systems based on human biology, for example, organoids, organ-on-chip, 3D tissues, and so on. And the main aim is to prioritize investment in research in India on these technologies. So we've seen that the use and knowledge of these technologies is scarce in India, and we aim to provide training and education to researchers through grants, workshops, lecture courses, newsletters, and webinars. So this is our work. So far, we have published 3 white papers on developing adverse outcome pathway frameworks, developing systems biology research, and using MPS as a model system, all in the Indian scenario. So we have also published 1 perspective paper and several popular science articles. We conduct monthly webinars, and this is a part of that series with scientists from all over the country. And we have periodically conducted multi-stakeholder roundtable meetings. We also have a quarterly newsletter. And recently, through the India Bioscience Outreach Grant, we have a virtual lab series called Back to the Future. So all of the links for these are present in our link tree, which we will be sharing in the chat box. Please do subscribe to us for future updates in recent advances in this topic, this methodologies. Yeah. So with that, I would like to pass it on to Dr. Vinod Scaria to take his talk for today.
Thank you very much, and hope the slides are visible and I'm audible. Yes, so over the next 30 odd minutes, I will give a brief overview of our journey through genomics and how you could walk from personal genomes to populations and from populations back to individuals to impact them in their healthcare. Now, before I start, of course, there is a bit of mandatory disclosures to make because we do work with a number of commercial organizations and they benefit both monetarily as well as in many other ways through these interactions. Now, the basic premise of my talk starts from around 2003, and I'm sure everybody in this webinar would appreciate that is when the human genome sequence was announced. Now, that was a very remarkable feat for humankind because it's for the first time that the entire alphabets of the human genome, amounting to approximately around 3 billion of nucleotides, could be deciphered, or for that matter, could be read. Now, this involved a very large international collaboration, which involved hundreds, if not thousands of individuals, both scientists and support individuals spanning multiple countries and approximately amounting to around 2.8 billion US dollars. Now, unfortunately, India was not part of this initiative because we could not foot the bill of sequencing the human genome or part of the human genome. But nevertheless, our neighbor China was indeed part of this initiative, and the Chinese researchers sequenced approximately around 1% of the human genome. Now, what really happened after 2003 was even more remarkable in many aspects, that it's so a slew of technologies, what today we call as next generation sequencing technologies, which would mean that the cost of sequencing would dwindle very rapidly, and in many ways in an exponential scale. To give you a perspective, this is essentially how the cost of nucleotides have dwindled in a log scale, just to keep this in mind, over the last 2 decades. And now what this would mean is that a number of genomic programs would come across the world, not just of humans, but also of animals, of pathogens, of plants, and so on and so forth. But nevertheless, the real question was how, as a country, we could actually monetize and we could take part into this genomic revolution. And that incident happened in 2008 and 2009, when India, for the first time, announced the human genome. And the premise of doing this is sort of exemplified in here, and this is a slide from way back in 2008. And what essentially the slide shows is essentially on the top part, the admixture plot of the world, the data which came from the Human Genome Diversity Program. And the bottom panel essentially is the whole genome sequences that came or that were available in public domain in 2008. So what you would see from here is quite significant in terms that almost all genomes which were available in 2008 came from quite less admixed populations across the world. But the most admixed populations across the world, which is from the Middle East to Central and Southeast Asia, largely did not have a genomic representation. And to put numbers, this encompasses approximately a third of the humankind as we know today. So in 2009, we put together one of the first sequences in the country. And the idea was to essentially not just sequence the human genome, but also to be able to learn the tricks of the trade, not just in terms of the assembly, but also in putting together an elaborate ways and means to analyze human genomes to understand eventually what is potentially functional and what could be really impactful for the individual in terms of his genome sequence and its healthcare benefits going forward. So in 2008 of December 9, this was announced in the Parliament by the Honorable Minister of Science and Technology, Shri Prithviraj Chavan. And this was later published in a peer-reviewed journal, as you would also see here. And this also provided us an opportunity to work with a fantastic set of collaborations. Dr. Sridhar, who has been a collaborator ever since, and fantastic students who now have moved forward across the world, evangelizing genomics in their own ways. Now, of course, one genome is not just what is required for this large population. So we went ahead and collaborated quite extensively with other countries across the world, also doing the first Sri Lankan and also contributing to the first Malaysian genomes. We were also one of the founding members of the Pan-Asian Population Genomics Initiative, which is a flagship program under the Human Genome Organization. And today we hold one of the largest databases of genetic diversity in this part of the world, including for South Asian populations encompassing around 1,200 odd individuals, for the Middle Eastern populations encompassing around 2,000 odd individuals, and much more recently for India with a pilot 1,000 odd whole genomes as part of the Indigen program. So the real question is not anymore whether we can do human genomes or whether we can collaborate across our boundaries, but largely, how are you going to impact human beings using this technology and using the analytical approaches and the data resources that you have access to? And that comes back to one of the areas where we could really make a huge impact. And just to keep in mind that this is just one of the areas where we could make a large impact, and that's for rare genetic conditions. Now, while they're called rare genetic conditions, in many ways they are not essentially rare. I'll come to that to the end of my talk. But nevertheless, since they are classified as rare, many individuals are not very aware about these conditions. Not even clinicians are in many ways aware about these conditions. And that would essentially mean that many of these individuals go through multiple rounds of physician visits, multiple rounds of misdiagnosis, and they lose a considerable amount of time before they can even get to a diagnosis. Now, to put numbers in perspective, it is estimated that approximately around 300 million people in this world suffer from one or the other genetic condition, rare genetic condition, and of them, around 70 million people in India alone. Now, that is really, really a large number, and that's what we wanted to also tap into and make a difference. And of course, doing such genetic studies in India also comes with an advantage, and the advantage being that India is an extremely large population, which encompasses today around a sixth of the global population. And unlike the rest of the world, our population is stratified into smaller groups. They're not really outbred population, they're endogamous groups, and it's estimated that there are around 4,000 odd well-defined endogamous groups in the country. And much more importantly, we still have a close-knit family structure. Everybody in this webinar could trace where their ancestors are from, where the grandparents are from, and that essentially means that you can start with a three-generation pedigree. Many regions of the world cannot actually do this because they have been displaced by famine, by multiple world wars, and therefore they may or may not be able to trace their ancestors, and therefore that is an advantage in a country like India, because you can essentially get an extended pedigree to do everything that you want to do in genomics. So in 2015, with my colleague, Dr. Sridhar Chivasubbu, we started this program called Guardian, and Guardian stands for Genomics for Understanding Rare Diseases India Alliance Network. And the idea of this program was essentially very simple, that they will work with clinicians who see undiagnosed genetic conditions in their clinics. They would use a very standardized process of referral, which involves a consent process, sample collection, and we would put them all for genomic analysis. And whenever we find the genetic mutation, we would get back to the clinician with the information so that he or she can act upon it in conjunction with the family. Now, of course, in such a large program, or of course in any genetics program, that necessarily means that you would not be able to get back to the clinician in all the times. And that is essentially when you want to step back into the wet lab or to the experimental lab to do a bunch of experiments, which might take a lot of time to actually prove that what you find is essentially what is pathogenic and what is actionable. And after that, or we reach to an evidence level that we can act upon that information, we actually go back to the clinician and inform so that he or she can act upon that information. Now, of course, to do this, you need to also educate clinicians and practically everybody in the value chain so that they can understand what this information means and what it would entail to act upon this genetic information. So over the last 7 odd or 8 odd years, we have built this network to encompass today around 300 odd clinicians from around 70 clinical and teaching centers across the country, making it one of the largest programs for rare disease research in this part of the world. And this program is entirely funded by CSIR through multiple different programs. So in 2019, before the pandemic struck, we had actually put together this paper where we show that how we can actually take the program from collaborations to research to diagnosis to actually empowering people and institutions through policy implementation. And as we know today, we have a national rare disease policy, which sort of creates an epic set of guidelines for interventions, for research and treatment of patients with rare genetic conditions. Now, I will give you a couple of examples on how this program works and how this program is a bit different from other programs across the world and also across the country. And this is one such family who was in news, I believe, sometime in 2015 or 2016. They petitioned the President of India to euthanize 6 out of their 8 kids. And of course, you could think of the plight of parents who would petition the President of India to euthanize their own kids. And it's largely because these children have a progressive neurological disorder, as you see here in the clockwise direction from the youngest to the oldest. At birth, they're pretty much normal, or they're perfectly normal until they reach around 3 to 4 years. And at 3 to 4 years, they eventually lose their milestones and end up in a vegetative state, and eventually they die. And of course, multiple rounds of diagnosis or attempts to diagnosis have been made, but nevertheless, there was no molecular confirmation, nor did the family have any hope that this is possible. So of course, since this was in news, this reached out to us, and we could actually make a diagnosis using genomic approaches as megalencephalic leukoencephalopathy with subcortical cyst type 1, or in short word, cystic megalencephaly, a genetic disease extremely rare in nature, which is caused by mutations in a gene called MLC1. Now, of course, this is just one family, a bunch of children in that particular family. We made the diagnosis. So what? What we did was essentially not just stop with the diagnosis. We actually went back to the community and asked a very simple question. If this disease was extremely rare, can we understand the prevalence of the disease in the population? Now, this family came from a very close consanguineous family called the Nalband, and the Nalband trace their origin from a different part of the world in the Middle East. They came with one of the invading armies because they had an extremely valuable skill of making horseshoes, and they stayed back in the periphery of the ruling families or the ruling communities, and then therefore formed this extremely consanguineous community called the Nalband. Now, screening a few hundred individuals, around 100 individuals, what we realized that even more remarkable, that the carrier frequency for this particular genetic mutation was extremely large, amounting to approximately around 28% in the close consanguineous community. Now, of course, you're in genetics, you would realize that a 28% of a carrier frequency in a close consanguineous community would essentially mean that 1 in every 300 to 1 in every 400 children that would be born with this genetic condition. So we actually went back to these communities and we traced additionally another 4 individuals with an extremely rare genetic condition in that community. And we could offer molecular tests for many of these families. So in summary, you could take from the case referral in 2016 to a molecular diagnosis to a publication which essentially shows the burden of this disease to actually a diagnostic in the market, not just benefiting just one individual or one family, but largely which could benefit the entire community so that they can avail carrier screening and, of course, with evidence, genetic counseling, and if possible, prenatal screening, so that you can eliminate this disease from occurring in the community. And to do this, of course, we work very closely with the All India Institute of Medical Sciences, Professor Shefali Gulati. But another important component is the need to work with social organizations. And in this particular case, we work with the Human Welfare Foundation, which still works among the community and helps the patients. And that's really important going forward, because it's not just the solution that is offered by technology, but also the social connect, which ensures that the benefits from the technology is continued, or the benefits from the technology is available to not just the family, but also the extended community around the family. Now, of course, this is another such case, and this case came from the Christian Medical College in Vellore. And this individual, a very well-educated businessman in his 20s, late 20s, presented with recurrent fever and joint pain spanning many years, and to that matter, around 5 years. And this individual have gone to multiple clinicians, multiple hospitals, got admitted, has taken practically every course of antibiotic or every combination of antibiotic, including anti-tubercular treatment. But nevertheless, he did not have any benefit with any one of these treatments. So he presented to the Christian Medical College in Vellore, where the clinicians suspected that this could be a genetic condition. And one of these genetic conditions are called familial periodic syndromes, fever syndromes. And one of the commonest familial periodic fever syndromes is a disease called familial Mediterranean fever, but nevertheless, it's not a disease which is very prevalent in this part of the world. Of course, by doing genomic sequencing, we could figure out that this is indeed a case of familial Mediterranean fever. We were perplexed because it's not a disease which is very prevalent in this part of the world. Therefore, we went back and asked this question: is it a possibility that this individual could trace his ancestry to the different part of the world, which is the Middle Eastern basin? And using genomic data, in fact, global genomic data, we could establish that this individual indeed is part admixed and indeed had a component of the genome which traces his ancestry from a different part of the world. So genomics can actually prove the molecular basis of a disease and also prove why that molecular basis of the disease could be explained in a particular situation with respect to the ancestry of the individual. And now, if you really think that these are abnormal or rare occurrences, you might actually be wrong, because there is a large number of individuals today in our community, and they are Indians in all aspects, but nevertheless, they trace their ancestry, at least genomically, to a different part of the world. And therefore, they would also have genetic diseases which are prevalent in a different part of the world. Now, since over centuries, they have been Indians, and they're in all aspects culturally and physiologically in all aspects Indians, but nevertheless, they do have genetic diseases which run in families. And since they're minority populations, they are usually not diagnosed because these conditions are rare in this part of the world. Now, this is just one example to show that there are individuals with a genetic component from different parts of the world. But we should also keep in mind is that there is also a much larger diaspora of individuals today across the world who trace their ancestry from India. Now, their ancestors have migrated from India, of course, as part of trade, as part of business, as part of slavery, but nevertheless, they are now spread across the world and have genetic diseases which are very prevalent in some communities in India, but now they have the genetic disease in a different part of the world. And in many cases, they remain undiagnosed or underdiagnosed, and in many ways not treatable. So that necessarily points out to the need of collaborating extensively, not just within the country, but also across the world, so that humankind can benefit from the genomic advancements. Now, having said that, we should also realize that the paradigms in genomics are also shifting very rapidly. Now, all that we have heard about genomics is based on the principle and concept that the patient will come to the clinician, and the clinician will diagnose the patient based on a set of signs and symptoms and history and a pedigree, offer a genetic diagnostic test, and the genetic diagnostic test will confirm that the individual indeed have a genetic condition. And that is essentially what Guardian was all built upon, that a clinician will diagnose the patient and the genetic tests will confirm the molecular basis of the disease. Now, just to take a step back and ask this question, now, if there are genetic diseases in the population, can we now prevent these genetic diseases from even happening, or even before they become a patient and they go to the doctor? And second point, you also need to keep in mind is that we started with a primary concept that clinicians are unable to diagnose many of these genetic conditions because of the lack of awareness. Now, if the clinicians have a lack of awareness, it is very unlikely, therefore, that they will have a very good estimate of what are prevalent genetic conditions, because they are essentially going to put as prevalence what they see and what they can understand. But what if they did not see or they did not diagnose? Can we identify such individuals with genetic conditions that clinicians are not really aware about? And can we really turn it upon itself, the concept that you can teach clinicians on what to go about in terms of prevalent conditions? Now, to do that, you need two things. One, you need to have a methodology to sequence individuals at very large numbers. And you also need to have evidence to be able to interpret the genomes of individuals without the clinical information. And once you have done that, or when you are able to do that, you can actually go back and ask this question, what is really prevalent in the population? And that is today possible thanks to advances in genomics that enables genome sequencing to be affordable and accessible. And at least by early next year, we're thinking of a $100 genome. That means that a significant large number of individuals could now access whole genome sequencing, even in an asymptomatic state. Like you and me, who are apparently normal, apparently do not have a genetic condition, could now access whole genome sequencing. And with the evidence accumulated over years, we could now ask this question, can we act upon the genetic information? And as I said before, that is also possible today because genomic sequencing technology has advanced to an extent that it is far more cheaper in the future. In a few months from now, it's going to be cheaper and far more accessible and far more faster to actually do a whole genome sequencing than do any other genetic test. It's also reached a point in time when whole genome sequencing is faster than a whole exome sequencing. And we anticipate that in a few months from now, the cost of whole genome sequencing and whole exome sequencing would pretty much remain the same. And that would necessarily mean that everybody could access whole genome sequencing. This would have a lot of implications because whole genome sequencing is all genetic tests put together. So you don't need to do a FISH, you don't need to do a karyotyping, you don't need to do an MLPA, you don't need to do a repeat prime PCR. You can do just one test, and that is whole genome sequencing. And being a fast and accessible test, which would mean that you would have a lot of implications going forward in clinical settings, like, for example, sick children in neonatal ICUs. Number of centers across the world are doing that now. Familial genetic conditions where you have no diagnosis. For prenatal genetic screening, when you have a family history but no identity for the molecular mutation. Now, this could really, really change the way we could look at medicine in the future. And much more importantly, by using population scale data, you can actually go back to clinicians and educate them about diseases that they don't apparently see. And this could really make the diagnosis of some prevalent treatable genetic disorders much better, much more efficient. So, of course, for that, you need to have a population scale data. And fortunately, in 2019, with my colleague Sridhar, we put together Indigen, which is one of the pilot programs for population scale genomes, which involves around 128 odd individuals, cosmopolitan populations across the country. Now, of course, with this genomic data, you could glean a lot of information, which are quite very valuable to clinical interventions. Now, of course, you could look at treatable genetic conditions. You could look at pharmacogenomics or variants which are associated with adverse drug events. You could look at cancer susceptibility variations in the populations or subpopulations, and you can also identify population-specific disorders. Now, of course, this is not just a hope. This is what the reality is, and the next few slides is essentially showing why this is important and why this is possible to be done on even just 1,000 genomes from India. Now, you would also need to realize that 1,000 genomes for a billion population is a very small number, but nevertheless, it would have enormous implications because 1 in 1,000 in India is essentially 1 million people, right? So even 1,000 genome could, of course, make a lot of difference. But of course, the hope is that we will be able to do many more thousands, if not hundreds of thousands of individuals in the years to come by. And as I said before, to look at whole genomes, which are apparently normal, you need to have evidence, which is cataloged in a very systematic fashion. And of course, now there are global guidelines which are followed for evidence evaluation. And for variants, we popularly use the ACMG and the AMP joint guidelines for interpretation of genetic variants, which classify variants into 5 classes as pathogenic, likely pathogenic, benign, likely benign, and VUS. But of course, you cannot really go back to a genome and evaluate every variant. You need to do the other way around by first consolidating the variants which are available and then asking the question, how many genomes have it? So you need to have very consolidated, comprehensive evidence resources. And this is one we created very recently called BRCA Indica for the BRCA1, BRCA2 variants. We worked with clinicians across the country, oncologists who have done BRCA1, BRCA2 testing. We compiled that information. We have also taken information from public domain databases. We have also taken information from the Indigen datasets. We went back to literature and evaluated every one single of these variants for evidence whether they are pathogenic or likely pathogenic and therefore actionable. And today we have probably globally largest data set of variants compiled and curated by the ACMG-AMP guidelines, which can now drive the genomics for BRCA1, BRCA2 hereditary breast and ovarian cancers in the country. The other missing piece is essentially the way to communicate information back to the clinician when the clinician requires that. And this is very important because nobody is going to step into a clinician's office with a hard drive of a whole genome. And you cannot expect the clinician to now be able to download the VCF file, pass through the VCF file, and figure out what is really wrong with the patient. And more importantly, at this point in time, the evidence is growing very rapidly. In other words, it is almost doubling every 3 years. Now, that would mean that a report at this point in time is probably obsolete the moment it has been released, because it doesn't really factor in emerging evidence in the future. So you need to have a genomic system or a digital system which can enable access to the clinical information in a secure, privacy protected, and a point of care, but nevertheless clinician-enabled fashion. If you really look at other paradigms which has happened across in the digital world, today we have a credit card or a debit card which we can swipe and get cash wherever we want. We have UPI-like systems where you can transact on your mobile device. Now, the real question is, can we do this for genomics? And of course, we built one prototype app which was implemented for the indigent program and therefore called the Indigenome card, which 1,000-odd individuals today carry. But of course, going forward, we need to have a much better system. And that is what we built with an organization called Zifo. And this is a commercial version which is available for any genomic organization which wants to implement this. So it's essentially a UPI-like interface for genomic information. And it's called Pocket Gene. You could find more information on this website. Now that you have a digital ecosystem, now the really big question is, how do we really go back and identify and teach clinicians how to implement genomics? So this is one of the two examples that I'm going to talk about. And this example is from the Calicut Medical College. I'm an alumni from that institution. We worked with my teacher, Dr. Geeta Govindaraj. And the idea is that from genomic information, we found that primary immunodeficiency disorders are quite prevalent in that population. We went back, set up a center, a specialized clinic with diagnostic support, also funded by the government and an NGO. And what we have identified was a large cohort of individuals who were previously undiagnosed for primary immunodeficiency conditions. And being in a teaching hospital, you have an advantage that you can propagate this information to the future generations of clinicians. And over 3.5 years, we have a large number of people who are on treatment for using IVIG. We have around a dozen individuals who have undergone hematopoietic stem cell transplantation, which is a cure for the condition, and many, many more families who access genetic diagnosis and prenatal genetic counseling and prenatal screening so that they can prevent the recurrence of the disease from happening in the family. And of course, there's another condition. This is a repeat expansion mutation in a very small community in southern India. We identified this pentanucleotide expansion mutation, which is a founder mutation. And with a very cost-effective genetic test, working together with Dr. Radha Mahadevan, who first described this genetic condition in that small community, we expanded this community screening to encompass around 434 patients characterized. And today we have a very cost-effective genetic diagnosis, which can be implemented in an approximate community size of around 5 lakh individuals. Now, to also keep you in perspective, this genetic disorder was molecularly characterized for the first time in 2019. There are only 13 cases reported in literature. And in a small community in India, we have 400 odd patients molecularly characterized for this genetic condition. And for that matter, this genetic condition is treatable. Now, you could also look at other treatable conditions because early diagnosis can actually make a difference for these children. And we could pass through indigent data with evidence in hand to understand what are the treatable causes of intellectual disability. And we could actually understand what are the very prevalent treatable conditions. And the most prevalent ones would be phenylketonuria, Wilson's disease, citrullinemia, so on and so forth. The real question is, of course, the prevalence is important. What can we do about it? So my colleague, Dr. Binu Kumar, essentially set up his program on Wilson's disease, which is a treatable cause of intellectual disability and a treatable genetic disorder, an extremely prevalent underdiagnosed genetic condition. He set up working towards it by working closely with a large number of gastroenterologists, creating one of the largest consortia in India for Wilson's disease. He created a very effective, cost-effective panel for diagnosis of Wilson's disease. And of course, when the patient numbers and samples go up, you also need to have resources which can interpret this genetic information. So Wilson Gen was created, which today is the most comprehensive genetic variant data set, all confirming the AIC-CCMB guidelines for Wilson's disease. And going forward, the idea is to implement ML and AI. So we have a Wilson Gen AI, which is under publication at this moment, which can accurately classify the pathogenicity of genetic variations without the need of going to experimentation. You could also look at very prevalent genetic conditions which are treatable. And one of the examples is familial hypercholesterolemia. 1 in around 200 odd individuals would have familial hypercholesterolemia. And in many ways, people who do an LDL test can actually figure this out. If you have more than an LDL of 250 or more, we are building this collaboration at this moment, working closely with biochemists who are doing LDL testing. And we offer a 3-gene test, which is very quite affordable and accessible so that every single of these family members could be screened and therefore be treated very effectively. You could now extend this to, for example, pharmacogenomics. And this is one such example for 5-fluorouracil, very commonly used anti-cancer drug. All solid tumor regimens have this. But of course, it has a very high level of adverse events in the population, estimated to be around 8 to 20% in different studies. You could use genomic data to figure out what is prevalent in the population, go back and validate that in a small cohort of individuals, and roll out a very cost-effective 4-variant test for 5-fluorouracil today, which oncologists can afford, patients can access it, and therefore can make a lot of difference in accurate and precise treatment of cancers. I think I'll stop there by summarizing to say that we work with clinicians with genetic conditions through the Guardian program. We offer a genetic diagnostic test, many genetic diagnostic tests which are prevalent in the population through a separate program called GOMED. I have not told more about it. It is run by my colleague, Dr. Mohammed Farooq at IGIB, another clinician scientist. We also educate clinicians through multiple avenues, online and offline. And we also established one of the first fellowship programs for genomic education for clinicians through the Sanofi Genzyme Fellowship Program. We work closely with the industry through commercially making many of these tests accessible and affordable, and can have a far reach to the clinician and patient community. We also work on genetic diagnostics and treatment for genetic diseases, because that's essentially the holy grail for all of the genetic conditions. And to do all this, what we do is to focus on the center P, that is participation, and that participation can enable genomics to be predictive. You can prevent genetic diseases from occurring in the community and families. You can make genetic diagnostic very precise and treatment very personalized. I think I'll stop there and take questions. But before that, let me acknowledge a lot of people who made this possible. A fantastic set of students in the lab and a lot of people who have actually moved out of the lab. Fantastic collaborations with Dr. Sridhar's lab at IGIB, who has been a long-standing collaborator for now close to around 15 years. Fantastic collaborators from across the country and across the world through multiple genome programs. And not the least, but the most important is funding, which has happened consistently through the last 1.5 odd decades through multiple different programs from CSIR. And for people who are really interested in learning more and they want to implement genomics in the clinical settings, there's a course that we are currently running for pharmacogenomics. You are invited to participate in it. It's a free course. It's an international course with teachers from across, or faculty members from across the world, and of course also participants from over 25 countries. And of course, if you have your colleagues who have children who are also now interested in genomics, we have a course in bioinformatics for school children, and I'm sure you would pass this word across. Thank you very much, and I can take questions.
Well, thank you so much for that wonderful talk. We do have a lot of questions coming in, so I'm just going to start reading them out. Maybe you can stop sharing your screen, sir, and then we'll just pin you so that everyone can see your video. Okay, so the first question is from Madhavan, and he's asked, how can prenatal screening be helpful in find the rare disease in advancing the effective treatment and diagnosis?
Yeah, so there are many genetic conditions which are familial in origin. That means that the parents in an autosomal recessive condition, the parents would be carriers for the genetic disease. The parents would be apparently normal. But when the alleles come together, there is a chance, 1 in 4 chance for a recessive condition, that the child will also develop the disease. Now, prenatal testing essentially means that you can test the fetus even before the child is born. And therefore, by the genetic counseling, which goes before the testing, and genetic counseling, which goes after the testing, the parents can decide the fate of the fetus, whether they want to have the child born with the genetic condition, or they can terminate the pregnancy. Now, that is the option that is available based on the evidence level, and this is applicable only for variants with high evidence level and for high penetrance. That's just an option so that the patients can make the decisions what they want to go ahead with.
Okay, the next question is from Shrikant. Can one analyze their genome using your developed tools in a privacy protected manner? And if yes, then which portals should one use?
Yeah, so there are multiple portals where you could use and analyze genomic data, but many of them may or may not follow the global standards for security and privacy. And that's why we developed Pocketgene as an approach. And the idea of Pocketgene is that it is essentially like your financial transaction system. You have money in your bank, and that is in physical formats. But when you transact using a plastic card, which is your debit or a credit card. Now, when you transact using a debit or a credit card, it doesn't mean that the money from the bank moves into your ATM. It doesn't happen that way. It happens on a digital platform and a ledger system, which means that the transaction has been authenticated, it is secure, and it is private. And this is essentially what Pocketgene also implements. So Pocketgene, or the card, doesn't have any data, doesn't have any information. The card authenticates with appropriate permissions the transaction of that information to happen from the physical form, which is a genome, which is residing on some server, to the point of care, which is a clinician who requires that information at that point in time. So that's essentially the concept of Pocketgene as a secure platform to hold on genomic information, interpret genomic information, and make it available on authentication to a clinician who requires it the most.
So the next question is, can genomic, I think it's familial disorders, here it's written familiar. So can genomic familial disorders be found out by this study?
Absolutely. So what we do in Guardian is essentially to do this over and over again for multiple, thousands of families who have now benefited from the diagnosis of a genetic condition by looking at their genome sequences.
Does India plan to have guidelines similar to ACMG and AMP for calling variants pathogenic?
Yeah, so that is a very interesting question, because see, guidelines need not be a national based guideline, because it doesn't serve much of a purpose unless there is a specific need in that particular population. Guidelines are always better to have an international level, because that also enables interoperability of information. So you don't want a pathogenic variant in a different population to be called pathogenic in Indian population and vice versa. It's always important to have the dialogue. If you find a pathogenic variation in your population, the rest of the world will also benefit from that information, and the vice versa, where a pathogenic information in a different population could also benefit patients in your country. You would always need to have a global consensus for guidelines, because that is beneficial for everybody. Each country having their own guideline, unless there is a very specific need or a very specific situation, is not a good idea going forward.
So there's a follow-up to the question about genomic familial disorders. They've asked, how can it be treated, and is stem cell therapy related to this?
Yeah, not all genomic or genetic diseases are treatable, but there is a subset of disorders which can be treated either by a small molecule, either by an enzyme, either by preventing the toxic part of the regular food that you take. Like, for example, you could have food which is deficient in particular amino acids, where the amino acid is cause for genetic disorder. You could also have, for example, blood disorders, hematopoietic stem cell transplant, which is also known as bone marrow transplant in common terms, which can actually transplant your bone marrow with a normal functional bone marrow and therefore replace your blood and blood products. Now, there are multiple different ways for it. Not all genetic disorders are treatable, but a significant small subset of genetic disorders, especially the prevalent ones, are today treatable.
Okay, the next question is, why is newborn screening so uncommon in India, at least for treatable genetic disorders, and what are the current limitations in its implementation?
Yeah, so newborn screening is not very popular in India, largely because there is a lack of awareness among patients, clinicians, and hospital or healthcare systems. One of the first newborn screening program in the country was implemented in the state of Goa, where they looked at a subset of genetic disorders which are prevalent. Now, the problem with prevalence estimate is also because many of this data until very recently came from the Western population. Now, the Indian populations might have difference in prevalent conditions which are treatable, and therefore there used to be a mismatch. Now, Kerala is one of the states which has actually implemented a statewide newborn screening program in government hospitals. So every children who are born in a government hospital goes through a newborn screening program, not just for genetic disorders, but also for many other treatable genetic or non-genetic conditions, including, for example, hypothyroidism, hearing loss, so on and so forth. Now, the real value in newborn screening is that once you have screened the individual, you can treat the individual early on so that you could have a perfectly normal child living to an adulthood as a perfectly normal individual contributing to the society. Now, awareness and leadership is what is really missing in newborn screening programs. And apart from that, the evidence on what is treatable and what is preventable and how prevalent are they are also important. And population scale genome programs are essentially going to make a difference there, because that's essentially going to tell you what is preventable and what is prevalent. Now that from indigen we know that Wilson's disease or primary immunodeficiency disorders are very prevalent, they're also treatable, would also provide the evidence that you can add these to the newborn screening programs. The other, of course, genetic cause that can be screened is hearing loss. You don't need a genetic test to screen for hearing loss. You can have much better tests, and therefore you could also implement them. So the point that I'm trying to make is that newborn screening doesn't always mean that it is a genetic test that you implement. It is a screening test. It could be a biochemical test, for example, LDL for familial hypercholesterolemia. It could be a physiological test, for example, for hearing loss. It could be a genetic test, for example, sickle cell anemia, thalassemia. But nevertheless, the idea is the endpoints are genetic, and they are also treatable or preventable.
Now, there is someone who is studying pharmaceutical chemistry, and they would like to know how they can contribute to this field. I think it's a broad question, but...
Yeah, so of course, there are many ways you could contribute to the field. As students or young researchers, one of the things that we teach them is to go back and evaluate variants according to the AIC-CCMB guidelines. There's an extensive course that we run with 16 odd modules and a few thousand odd individuals from across the world participating. And this is, of course, an opportunity for you to learn about the guidelines and also help the community build upon the variants, which could help a lot of more patients.
The next question is, can autism be treated with the help of genomics?
Yeah, autism is a broad spectrum of disorder. It's not just one disorder. It's a broad spectrum of disorder. Therefore, we call it autism spectrum disorders. A subset of autism spectrum disorders which have a genetic cause could actually be treated. They present as autism, but actually the underlying cause is a genetic disorder. If you have a molecular diagnosis for that subset of autism spectrum disorders, of course, they are treatable, but not all.
Okay. The next question is, how can genomic data let us know about the many genes involved, and how can you precisely narrow it down to one specific gene?
Yeah, so the principle of genetics is that you have a heritable trait with high penetrance, and therefore you would want to look at individuals also within the same family or the same community who share the genotype and also share the phenotype. So the narrowing down process is not really a black box or hand waving. It is essentially based on standard principles of Mendelian inheritance and filtering the variations which follow the standard principles of Mendelian inheritance, and actually going back to proving that that variant indeed caused that molecular phenotype. So it is not a hand waving exercise. It's actually a very rigorous science that goes behind establishing a particular gene or a particular variant is causing a particular genetic condition.
Now, the next one is a follow-up question to something that was previously asked. So, are the pocket gene results only available to the clinician, and can the individuals whose genome is being analyzed, can they also access the analyzed data?
Yeah, so the pocket gene actually provides two tiers of information. Like, for example, if you are a carrier for a genetic disorder, it will tell you that you're a carrier for a genetic disorder. It will not tell you what variant or what genetic disorder. That variant information and the genetic disorder identity actually comes to the clinician, because you don't want individuals to act upon this information without adequate genetic counseling and clinical intervention and clinical decision support. So some information which is valuable is available. Like, for example, in pharmacogenomics, it will tell you that individual has a pharmacogenomic variant for a particular medication. It will not tell you whether it's associated with a lower dose response, a higher dose response, or you would want to change the medication. That is for the clinician to decide, and that is available to the clinician. But whether that you have a pharmacogenomic variant for a particular drug is available to the patient.
The next question is, the prevalence of spinal muscular atrophy is found to be more common in recent times, and parents are finding it difficult to afford the medicine, which costs about 18 crores. So do you think parents should do genetic screening before they get married or before planning a baby?
Yeah, so for many genetic conditions, the prevalence estimates in the population is pretty skewed. And that is largely because of two factors. That's largely because of clinician awareness and patient awareness. Now, many disorders that we see in headlines may or may not be as prevalent as they appear in headlines. Just because there is more awareness around a disease doesn't necessarily mean that those disorders are prevalent. Now, it's not to say that SMA is more prevalent or less prevalent, but also to say that you should not base your assumptions based on news articles. Just because a disease appears in news doesn't necessarily mean that it is prevalent, right? But nevertheless, the second follow-up question is that, of course, it has a very expensive treatment at this moment available and also marketed by a pharmaceutical company. There is, of course, differences in opinion between even clinical community regarding the utility of that particular drug, the utility of long-term use of that particular drug, and in terms of the cost-benefit aspect. But having said that, that are philosophical arguments aside, the point that we need to keep in mind is that we need more indigenous manufacturers, which can bring the cost down. We need to have better science and better evidence to suggest a particular molecule is efficacious or non-efficacious. And more importantly, we need to make decisions based on evidence on what is prevalent, what is effective treatment, and whether the treatment is affordable. So we cannot really see it just in the eyes of prevalence, just in the eyes of the efficacy, just in the eyes of the cost of the therapy. We need to actually look at a holistic fashion to see, given the prevalence of genetic disorders, given the subsets of this prevalent genetic disorders which are treatable, what is the real cost benefit aspect of treating genetic conditions? Now, this is a very unique situation because it also conflicts with many aspects of ethics and morality. Also because we are not an insurance-driven healthcare system. Our healthcare system is largely driven by out-of-pocket expenses. So at some point of time, we need to have a pragmatic way of viewing this and having discussion with all stakeholders involved, not just the patients, families, and their clinicians, but also diagnostic and therapeutic manufacturers, so that we can figure out a middle path for it. I'm not for any one side. I'm in for the discussions to happen so that we can have a pragmatic discussion and a middle path which is acceptable and so for everybody. Yeah, in terms of the third question, whether the screening should be made before or after marriage, it is a very tricky question to make. I think as adults, it is up to the adults to make that decision whether they want to make that screening before or after marriage. There are certain use cases where people have done this, for example, before marriage, like for example, the Middle East. There's some countries where thalassemia screening is mandatory for every college-going child or college-going adult, 18 years and above is an adult. They screen beforehand and they give this information beforehand, and they can decide upon whom to marry, etc., based on their decisions. So one thing that we need to keep in mind is that there are social traditions which you don't want to change, but you could modulate it using the evidence that you want. Give it to adults and let them make the decisions upon themselves.
Okay, the next one is, as they've written, it's a general question. Which will be more effective as a source of information for developing drugs, genomic, transcriptomic, or proteomics data?
No, so there is nothing that genomics or transcriptomics is better or worse. It is all dependent upon the case scenario and also dependent upon the disease that you're talking about. In many cases, you don't even need genomics to actually make a decision upon whether it's a good target or not. So let's not really try to compare and contrast different techniques and say that this technique is better or worse. I think what we need to look at is what is the best technique to solve a particular condition, rather than asking this question, what should I do, genomics or transcriptomics? Like I earlier said, for many genetic disorders, the screening test is not even genetic. There could be as simple as a cheap biochemical test or as simple as a cheap physiological test.
Okay, and our final question is, to do genomic sequencing for the whole family is a costly affair. So do you have to proceed with the whole genomic sequence to confirm the disease?
Yeah, as we talk at this moment in time, yes, genomic sequencing is a costly affair. Now, having said that, when genomic sequence becomes a $100 test, or in the future, a much cheaper test, probably $10 in a decade's time, I believe that every single human individual, it is probably going to be a right to have their genome sequence. We should not really make a decision based on the cost and the technology today. We should probably look at the technology and the cost effectiveness tomorrow. And more importantly, the benefit such a genome sequence can bring to an individual and to a society or a community. So in a decade from now, if we are all alive, probably all of us would have our genome sequence, or rather, all of us would be able to afford to have our genome sequence. And probably in the coming years, genome sequencing, like education, probably is also going to be the right of an individual.
Okay, so I think one more question is there. Please share any details about bioinformatics for school programs.
Yeah, you could visit my website, and the link is available on my website, or it's also on Twitter and LinkedIn. Or if you could just drop in an email, I would be more than happy to reply back.
Yeah, so unfortunately, that's all the time we have today for questions. So if anyone has any follow-up questions, As Dr. Vinod said, you can please reach out to him on his social media handles or on email. So thank you so much, Dr. Vinod, for this session. It was wonderful. We had a great turnout and we've had a lot of great questions. Thank you so much for giving us your time today. And
thank you very much. It's always my pleasure.
Yeah. And thanks everyone for joining. So this video will be available on our YouTube channel, on the CPHMS YouTube channel. So you can find it in the Linktree link that we sent. If you have any follow-up questions, if you'd like to discuss, you can reach out to Dr. Vinod. And we hope to see you again in the next webinar. Thank you so much.
Thank you very much.